traumatic brain injury. Michael Pierce, DC, DAC, MB, FAC, FN, was steered by his personal chiropractors to Palmer College of Chiropractic in Iowa, the fountainhead of the discipline. They were steeped in the traditions and science of the institution and helped him through the grueling education. Later, Dr. Pierce studied animal chiropractic and earned a certification and then studied the functional neurology of Dr. Frederick Robert Carrick. Dr. Pierce studied for years and earned the rare specialty in chiropractic neurology and treated much more complicated patients. Dr. Pierce is an assistant professor for the Carrick Institute of Clinical Neuroscience and the Rehabilitation, where he teaches molecular and clinical neurochemistry. In addition, he has been a lecturer for the Apex Energetics since 2009, educating physicians and other practitioners on the testing and clinical applications of functional endocrinology, immunology, and blood chemistry. Dr. Michael Pierce.
Thank you and welcome. I used to teach for the Carrick Institute and have been on sabbatical for a number of years. I went to cooking school and became a chef in 2012, and then I went to business school and dropped out in 2014, and so I had a kind of an interesting experience over the last few years so that I could earn some skills that would assist in helping brain patients and patients that were of chronic illness nature. So for us, traumatic brain injury really ties together all the conditions because it's very unique. All the conditions that we've looked at in this presentation today, they all have a certain pattern. If you've got a stroke, you've got certain blood vessels that are affected, and the watershed of those certain blood vessels downstream are affected. And those are pretty standardized sets by now in the understanding of neurology. If you have Alzheimer's, if you have Parkinson's, if you have PTSD, all of these pathways, all these patterns of dysfunction in the brain are patterns that are known, especially by the modern imaging. That modern imaging came to us in the decade of the brain. The National Institutes of Health in Bethesda, Maryland paid millions of dollars to universities in the decade 1990 to 2000. And that was the decade of the brain in which we studied all kinds of things and all kinds of people, from young people to old people, everything from brushing your teeth to reading a book to watching a movie, and understood that there are certain things we all share in common about patterns of how the brain fires in normal function and how the brain fires in pathology and disease. So that decade gave us a lot of information. That was the decade that was the real birth of functional neurology. So functional neurology was kind of invented by psychologists that were studying functional imaging, meaning as opposed to anatomical imaging, which is like an X-ray or an MRI of your brain that shows if a part of your brain is there or not there, if it's been co-opted by a tumor or a stroke. That's structural imaging. Functional imaging instead shows blood flow and glucose utilization and how different parts of the brain are firing, especially compared to normative data. What that means is if you've got a region of the brain that's not firing as well as it ought to, we'll color it a certain color in our reports, usually a color bluish in most traditions, to show that there's reduced frequency of firing of those neurons. In another type of functional imaging, or in all types of functional imaging, we would see other areas that are overfiring compared to normative parts of the brain. So you might find that a person has, in any disease, a certain region of their brain that is underfiring and another region that might be overfiring. And we'd color that red in most cases, although there are some different systems that might use yellow or green, but they all pretty much are moving toward using color to describe that one area is firing differently than another area. We want to compare that to normative data, and we do that with computers and data sets. So traumatic brain injury is different because you've got different vectors of impact. So a person could be hit from something above that fell on them from a shelf, and they might have a coup injury to this part of their brain and a contra coup injury here as the brain sloshes around from the impact. They might likewise hit a windshield, or they might just have a whiplash and not hit anything at all with their head and still suffer a brain injury. So traumatic brain injury represents kind of the weirdest, toughest, strangest, least consistent kinds of brain dysfunctions that you've looked at today. Because all the things you've looked at today have exhibited patterns. And those patterns have been discovered by folks that do SPECT imaging or fMRI imaging or EEG imaging or magnetic imaging of the brain. All those different methods that are functional that show the amount of activity of cells show us that there's patterns to each kind of illness. There's an identifiable pattern to Alzheimer's early and late. There's an identifiable pattern to misfiring in carbon monoxide poisoning. There's an identifiable pattern to Parkinson's disease. There's an identifiable pattern to what stroke looks like with different blood vessels that have been affected by a thrombus or a clot. But traumatic brain injury has this really strange quality, especially when patients have multiple traumatic brain injuries, when they've had an impact over here, and then a whiplash, and then maybe PTSD on top of it. PTSD can create some physical changes in the brain over time that look just like physical trauma, although maybe no physical trauma has happened to that patient, only emotional trauma. And I hate to say only emotional trauma because my whole point is that emotional trauma can be as bad as physical trauma. It can be as bad as a blow to the head, sometimes worse. And we can see that in imaging of functional nature. So let's take a look. I want to thank you to sarascan.com, who sponsored our booth and our talk today and who's provided a lot of the science behind what I'm talking about today. Fitforlife.org, not the Fit for Life that is a for-profit food product company, but Fit for Life, the nonprofit. I'm very excited to talk about that. We didn't get to have our chairman here today, Bill Schenken. He's a very famous accountant who brought the Colorado Rockies to Colorado. He did a lot of work in nonprofits. He's the top one of the top 10 fundraisers for Children's Hospital. So we're partnered with a lot of very strategic partners, and Fit for Life is trying to raise money to be able to pay for these expensive scans when people need a SARA scan or some type of SPECT imaging, and to pay for their care. We're also developing a model of immersion care, destination care, where patients can come to an actual campus and stay there for a period of time. Because right now, there's a difference between inpatient care and outpatient care, and there's this no man's land between them. So inpatient care is a hospital, and that's not really useful for chronic human illness because you don't need an intensive care unit if you have chronic illness. You need intensive care as a colloquialism, but you don't need IVs and what they do for acute care. Then there's outpatient care, which is what we all are familiar with. You go to a doctor, you go to a clinician, you go to a physical therapist, you go to all of you and psychologists, and you get care on an hourly basis. And some patients are too sick for that. So what we're working on is a model where we can actually have an immersion system where people come and stay immersed in care, immersed in treatment. They can get a lot more done in 2 or 3 days or 10 days or 90 days than they ever could in an outpatient model. So that space between outpatient and inpatient is the immersion residential campus type of thing. We're working on that. We're raising tens of millions of dollars for that project. And I actually got a chance to do that 20 years ago when I was just out of school and just beginning to really understand things and try to accomplish patient care. I was luckily hired to work at a center that was 2,000 acres with 100 beds and a commercial kitchen, which is one of the reasons I ended up going to chef school later on in my life to learn more about cooking for ill people because it's so difficult. And I'm working on a ketogenic project right now with regard to brain care. So let's go on to invisible disabilities. We've got to thank Wayne and the entire team here. This is an unbelievable thing. We were talking today about how much we need this kind of a meeting where we have chiropractic meetings, we have neurology meetings, we have psychology meetings and psychiatry meetings, and everybody goes and they all talk to each other and they never mix with each other. So we've got to do a lot more of this. So thank you, Wayne. This is just incredible. The NBCC for psychologists for continuing education, I've got courses approved by them in the past and by others. University of Phoenix is one of our sponsors. Many others, the Integrated Health Group, raise your hand, Doc, Integrated Health Group in the back there, and of course Dr. Josh Wallard of Sarascan and Fit for Life. I want to thank you. Doing marvelous work in stem cell therapy for the brain, Dr. Josh, so incredible stuff. So TBI and MTBI are varied and unique. As I talked about, there's all these different impacts. Different people have different genetics, like SNPs. They might have single nucleotide polymorphisms that change their metabolism, but do not actually code for disease. So as alternative practitioners or holistic practitioners, we're stuck in this limbo where the patients don't qualify for a genetic disease diagnosis, but they do qualify for an abnormal metabolism. So what does that mean? There's no diagnosis code for it. There's no payment code for it. There's no DSM-5 code or ICD-10 code, and there's no CPT code for paying for it. So how do we care for them? They fall into this limbo, and that's where we come in. They have unstandard impacts dispersed through the skull. You don't know how an individual might respond to an impact. Some people that have early sports impacts do very, very well, and in the rest of their lives, some papers show that they don't have much problems with concussion if they get conditioned as a child. Other children get concussions as a child, and they end up getting terrible problems with concussions later. Some people build an immunity to it, almost like a vaccination. It's like early concussions can be a vaccination for them, as if. And then we see many regions of the brain can be affected in disorganized patterns. So assessment requires, and treatment requires customization. There isn't a way to really have a standard boilerplate protocol for these patients, and it requires clinicians to be bold, and it requires them to take educated guesses and do therapeutic trials. Therapeutic trials lately have been poo-pooed a lot by scientists and by policymakers because you can't pay for them, number one, and you can't code for them, number two, and number three, it's hard to prove that they're efficacious. It takes really good doctors that are gathering data to be able to do that. And so that means if you're going to have customized care that's customized for an individual patient where you're following a theory of mind to try to take care of that person, you've got to gather data to check yourself to make sure you're not leading down a road that ends up being fraudulent or useless. So falls are primary in causes of TBIs. Take a look at this. Falls represent 35% here of TBIs. So if you've got a problem with a population of traumatic brain injury, it's very useful to consider the part of the brain called the cerebellum that's useful for coordination. It controls speed, timing, coordination, balance, and eye movements, not to mention learning and attention and a bunch of other things. So the cerebellum is something that's often not talked about because in the brain, there's this thing called a tentorium, and the tentorium goes across right about here, and it's kind of like a net. It catches the brain and holds it up and holds the brain in the fluid in this fashion, where there's also a falx that goes down the middle like this and separates the hemispheres. But this part of the brain right here, the tentorium, separates the upper brain from the lower brain. And there's this kind of adage in psychology and psychiatry that down below the tentorium is no man's land. So if you paid attention today, a lot of people will talk about therapies and assessments that are designed for the upper brain, the cortex, the surface of the brain that you can actually touch if you were to take off the calvarium, the cap of the skull, and you could touch that brain. If you could take away the dura, the sheath that covers it, if you could touch that actual brain, that would be the cortex and the lobes of the brain. You've heard of those before. That's the frontal, the parietal, the temporal, the occipital lobes. But the deep structures like the basal ganglia, the brainstem, and the cerebellum are considered no man's land because these are reflexogenic, these are animalistic, these are the reptilian parts of the brain. These are the areas that we consider that's not what separates us from the animals and the beasts of the field. That's the stuff that everybody shares, all animals, all mammals, all reptiles. We all share that same wiring. So it's not important. We should focus on the parts that are very cortical, that are very human, that involve meaning and music and science and language, all the important stuff. But our contention in functional neurology is that there's a bottom-up theory and there's a top-down theory, and both are important. Sometimes you have to fix the bottom stuff first before you can get to the top stuff. So for example, if you've ever had a patient with TBI or another illness of the brain and you're trying to rehabilitate them by starting with very high-level therapies, you're giving them cortical therapies that involve language and speech and meaning and nuance, and they don't have their eye movements down, and they don't have their balance down, and they can't touch their nose, you gotta start with that bottom stuff down there in no man's land and work your way up. In addition, if you have a person, if you know that falls are a major cause and motor vehicle traffic are a major cause, might it be nice if we could be more coordinated in our everyday activity? Make sure I don't fall off the stage as I talk about that. It's very important to make sure that as we drive and as we walk and as we climb stairs and as we do our daily living, that we can prevent falls by having a very healthy cerebellum. And that means doing a cerebellum exam. That means going to somebody that knows how to examine the cerebellum and doing our own cerebellum work, which I'll get to today. So ICD-10 codes are often irrelevant to real progress, and they don't address function. You have to qualify for a real diagnosis, and if you don't qualify for a diagnosis, you're not sick enough, get out of here, next patient. Does that resonate with anybody? Okay. So you can't establish a mechanism or affected brain circuits. Diagnosis codes are too generic. You have to be much more specific because each person's injury is very specific to them. Sometimes it damages a gray matter area that's very specific. Sometimes it involves a gray matter area that's very large. Sometimes it involves a white matter area that is a highway that carries communication from one area to another. And the treatments for all those are different. The pitfalls of rehab. There are some pitfalls in rehabbing people. These CPT codes, the procedural codes are out of date and not customizable. A lot of people don't get paid for it, so this becomes a cash practice. Many providers have left the third-party payer system for cash and neural fatigue's ignored. Patients are pushed too hard. This is a really important thing. And it's going to be our savior, actually, which I'll get to. The rehab may not be specific enough. We might be moving a limb when we might be needing to move a finger instead of a limb. Or we might be moving an entire arm when we might be needing to move just an elbow or isolating a joint. So all those refinements are significant. There's no paradigm for applying healthy brain areas to drive unhealthy ones. So did you know that in functional medicine and functional neurology, sometimes we actually target parts of the brain by using pathways that are intact? And that isn't talked about very much. Sometimes we drive a part of the brain that's very strong because it's like shooting pool. You bounce one ball off of another ball to get the ball to go in the pocket. So sometimes as we bank these shots off different parts of the brain, you have to use healthy parts and not just the unhealthy parts. That's a new concept that, again, is not talked about quite a lot in functional neurology. I'd like to mention EEG now, electroencephalograph and quantitative electroencephalograph and neurofeedback. This data can infer deep activity, but it really has limits. It can't visualize the cerebellum, for example, and can't visualize that no man's land. Where's the no man's land? Draw a line right there. And everything below it is the brainstem and the cerebellum and the eyes, and that's the no man's land. That's that reflexive area that involves eye movements and respiration and coordination and timing and speed. It's not the sexy stuff. It's not the parietal lobe and the frontal lobe where all the cool stuff happens, right? It's the basement. So this particular imaging system can show us down into the brain, and it can infer a little bit of depth, but not enough depth. It's great, and I like it. I'm not criticizing it. I'm just saying it's got its limits. So the real issue is dead neurons versus misfiring neurons. How do you know if a neuron is dead, or if a pool of neurons are dead, or if they're just mostly dead? Some of you will get that reference. I'm not completely dead, I'm mostly dead. Yes, good, very good. You win the gluten-free cookie, very good. So you can tell a dead versus simply impaired neuron by testing it. You've got to ask it to do something, and you've got to ask it to do something. Do you think you would want to ask it to do something at a very high demand rate or a very low demand rate if it was damaged? A low demand rate. Yeah, you don't want to give it a high demand. You don't want to ask it to, run back and throw a giant football. You want to be able to say, can you move your finger? Can you move your eye just a little bit? And we've done that in coma recovery work. I've been a coma recovery team leader and been able to work with these patients and do some of the worst of the worst. And in fact, my career, and some of you in the room, has been the most difficult patients where the typical protocols don't work, and we have to figure out why they don't work. And this is how we figure out why they don't work and what to do. This is the method. That method I talked about in an earlier slide called fatigability, which I'll get to, and this method of are they dead or are they mostly dead? Now, why do we care if they're dead or mostly dead? Can you rehabilitate a dead neuron after age 2? Well, we don't make a lot of new neurons in our brain after age 2. There's a couple of locations in our brain that we make new neurons after age 2, but mostly we don't. Now, stem cells may change that. But the point is, if we don't readily make new neurons after age 2, then what do we got to do? We've got to build plasticity among the nerves that are left and the ones that are left alive. So if they're left alive but not functioning very well, our job is to increase plasticity. How do you increase plasticity? Well, you guys have seen today, you drive that pathway. My question to you is, do you know how vigorously to drive that pathway? And what if you overdrive it? Has anyone considered that? We're going to talk about that now. We have to assess autonomic integrity for the tissue tested, which is to say vitals. If you do a brain exercise and the patient's heart rate increases, should you keep doing that exercise or should you take a break? Our theory is you should take a break. If you work on a patient and they have normal pupils and you do an exercise, whatever it is, I don't care if it's EMDR, I don't care if it's the limb movements that you saw earlier, I don't care if it's cortical exercises, and their pupils suddenly widen, inappropriate to the room. Should you stop for a moment and let them recover? Yes, that's fatigability of the autonomic nervous system. That may not show up in the patient's actual performance of the task, be it eye movements, limb movements, or cognitive higher functions. It would be an autonomic sign that would tell you. Now, you might say, and rightly so, that patient can just tell us they feel bad. But wouldn't you want to detect it before the patient can actually feel it? Because once the patient can feel it, now we've got an involvement of whole massive parts of the brain. We want to detect it before the patient can feel it, because blood flow has changed massively before the patient can feel a sense of nausea or a sense of doom or a sense of fear or a sense of anxiety induced by us, the rehabilitators. Yes? Okay, good. So we want to activate the patients with trivial inputs, something like smooth pursuits. We don't want to necessarily do the typical hospital procedure or the police procedure at the side of the road where we do pursuits like this, which is the way it's normally done. With these patients, we might do pursuits like this. Does that make sense? Which one would be more demanding on a patient that's got sick neurons? Is there a possibility or a probability that we might over-fatigue those neurons if we gave that person who had problems in their brainstem, which controls the pursuit mechanism, an exercise like this? And they suddenly throw up on you. Because it happens. Are you following? Yes. Okay, so assess for direct fatigue and the tissue tested. So one of the two ways to test for fatigue is the direct task. If I'm asking a patient to do a task like this, and they do 2 reps, and then they start skipping all around, or they eyes roll up in their head, or their pupil, or they do something like that where it's a direct task problem. The eyes can't do the task, they can't smoothly follow the target, so they start skipping around, or they miss the target, or they go up and down, or they suddenly shoot up in their head and look at the ceiling. Or they start looking around and they start looking at you. That's a direct sign of fatigue. They can't perform the actual task. The other sign, which I just mentioned earlier, is they can't maintain the autonomic function. Their pupils dilate, their heart starts to race, their face starts to sweat, their upper lip starts to beat up, their hand starts to tremor. Understand? That would be an autonomic sign of fatigue, not a direct sign, because they might be able to do that, clip along and say, this is great. Meanwhile, they start sweating and their pupils get huge. Is that good or bad? Thank you, very good. Okay, so you want to assess for autonomic fatigue by supplying the tissue tested to getting sweaty or nauseous, and you want to prescribe a precise number of reps. Do you think at this point with this fragile patient that 2 reps versus 3 reps might be significantly different, and that 20 minutes might be just destruction for them? Sometimes. And how do you assess it? You do the 2 things I mentioned. You look for direct fatigue of the task, and you look for autonomic fatigue. Every time, all the time, continuously, every single time you do anything to a patient. All the way from the first minute to the 20th minute. Because most standard payers pay 20-minute sessions, don't they? Right, yeah. Is that smart? Maybe not always. Let's go back to 1900, the neuron theory of Cajal. Ramon Cajal, the Spanish neurologist who used horseradish peroxidase to dye cells and show us the differences between certain neuron cells and other nerve cells in the brain. In 1900, this is the year that he developed the neuron theory that neurons require 3 things. They require fuel, which is a few things. It's not just glucose, it's a few things. But they require fuel, they require oxygen in the blood, and they require activation. They're like needy babies or needy little monkeys, and you may have heard of the monkey studies where if you don't give affection to these babies or monkeys, that they actually die of what seems like a broken heart. They don't get the stimulation they need. Well, neurons are just like those babies, just like those monkeys. They need activation. They need to be elbowed by nearby neurons that say, wake up, fire, you've got to be awake. And that activation can be either stimulation, where it's a message that says, fire, do something. Or it can be a nearby message that says, don't fire, inhibit. And so that neuron is sitting there like a child in school getting a stimulation, which basically says, shut up, don't move. And that neuron doesn't move, it doesn't fire. So activation can be either stimulation or inhibition, which is hard conceptually to understand. So we talked about the decade of the brain. Lots of tasks are involved. Medical studies show that it takes 17 years for new science discoveries in the literature to make it to med school curriculums. That's pretty awful. I write curriculums, and it's pretty awful. The neat thing about this is most of the tasks that we do in life are preserved in all of us, so that generally, if you do a task and I do a task like touching my finger to my nose, I'm going to activate the same brain pathways. If I use my right hand, I'm going to activate the same basic pathways in my right cerebellum and my left brain. We need to move toward balance between hemisphericity and brain localization and other theories. That's a little complicated, and I may get to that. We have to understand hierarchies. We have to understand, as I said before, that sometimes you have to have a bottom-up philosophy where you rehab the bottom reflexes first, like stability, balance, vestibular ability to level your head, autonomic function like eye movements and pupils and respiration and sleep and all those things, before you can develop those cognitive structures that are higher. So if you go after cognitive therapy and it doesn't work, consider backing down the brain and go back into that basement that I talked about earlier and rehab that area first. And don't forget about fatigability. 2 types of fatigability, right? Some skills precede others, and so must rehab, and that's different for each patient, but it has a general idea. This is a paper I published a couple years ago with a group, Dr. Brock and Yannick and me and Michael Powell, a bunch of these guys, and what we did is we did a review of all the information on non-drug, non-surgical assessment and treatment of people with TBI, and this journal is about to be indexed, and so we're waiting for that to happen, the Neuroscience publishers. It was a very interesting, very long paper, lots of authors, lots of work, and it goes through all those eye mechanisms, all those body mechanisms, all the stuff I'm talking about today and don't have time to explain. It's a 2013 paper in the journal Functional Neurological Rehabilitation and Ergonomics. So find the source of the problem. Use functional imaging, use evidence. This is not voodoo. SPECT imaging, high resolution with data processing. That's what we do now. The old SPECT was very low resolution and it didn't have data processing computers to understand what we were seeing. It was very grainy and very not clear. So today's is much better. QEEG and ERP, that's event-related potentials or evokes potentials. Those are task-related things, whereas EEG is more like you're sitting there at rest and they analyze your brain, where ERPs are they ask you to do something and they measure your brain waves. Comprehensive medical and family history, 90% of diagnosis comes from what? History taking. All medical diagnosis textbooks tell you this in the first chapter. 90% of diagnosis comes from the history. Validated paper instruments and neuropsych testing, of course. Toxicology screening to make sure they're not on drugs that you don't know about. Skilled functional neurological exam with data collection, that's very important. That's what I'm talking about today, is really understanding what area of the brain you're looking at when you see a person do a task. When you see a person's arm tremor when they do something, where is that in the brain? If you don't know, ask someone and really get help. Metabolic lab testing for functional medicine imbalances, that's a very important one. That's often missed, and yet people in this room have talked about it. I'm very proud of you and very excited to hear you talk about that. So this is just an example, a slide that shows all the complexity of those pursuit mechanisms. Pursuits are when you follow a moving target, and saccades are when you jump from a target to a target like this. Now, EMDR uses pursuits to follow a moving target. What if I had an injury where that thing fell off the shelf and hit me in the head right here, and it gave me a contra-coup concussion to my left cerebellum? And so I go to my EMDR doctor, and I have PTSD and a TBI, and I say, I want to do EMDR. I hear it really helps. And I love EMDR. I love EMDR. I refer to it all the time. I love it. And that EMDR doctor says, let's do a pursuit. And so when we do this, we know that the right cerebellum drives the eyes to the left, and the left cerebellum drives the eyes to the right. So that doctor may not know, with that injured left cerebellum from the contra-coup concussion that hit over here and hurt right here, with me so far? That person's doing this, what are we going to start seeing? Do we need to give equal therapy to the right cerebellum and the left cerebellum? Which cerebellum needs the therapy? Which one is injured? The left one. So which one needs the therapy? The left one. So do we need to do, if we know in neurology that we need to target the tissue specifically, we can't stimulate the whole brain, it doesn't get better. The genes don't turn on if you stimulate the entire brain. You have to be specific. Otherwise, we'd all just take cocaine or something and stimulate our brains to fire overall. It doesn't work to stimulate the whole brain globally. You have to stimulate specific parts for it to work. So if we have that problem, and we go this way and this way, when we go this way, our normal right cerebellum is firing, big deal, yawn. It doesn't need any help. It's fine. Which cerebellum needs the activation? The left one. And the left one does this, right? So we might say, well, gosh, let's do EMDR only to the right from the left cerebellum. Well, that's cool, but we're lucky enough that we have an inner ear system and a cerebellar system that makes us know that this action is left and left cerebellum, because we're looking at both of the canals in the ear. I can't explain it today, but trust me, the canals in the ear and the cerebellum and the anatomy all make it that this is all left cerebellum and this is all right cerebellum. So if you were making the decision about EMDR for me and I'm injured on my left cerebellum, do I need this exercise? Will this help me? If I need to isolate my left cerebellum? No, right? Do I need to do this exercise, which is which cerebellum? This is the right cerebellum. Is this going to help me? No. Which exercise do I need to do? This one, which is for the left cerebellum, up left and down right, that's for the left cerebellum. Now you've taken and solved that. Take your right hand, reach behind you, pat yourself on the back. You solved it, very good. Now here's the next question. Is it possible to overdrive that left cerebellum that's been injured now? I've got this injured left cerebellum, it's all messed up, and I've got this eye movement. Is it possible for me to do too much of the dose of that therapy, just like I could overdose a drug like insulin? Is that possible? Yes, it is. How would I look for that if I was going to fatigue that left cerebellum? I would look for two kinds of fatigue, wouldn't I? And those two kinds of fatigue would be the direct kind, which is they fail at the task. As that patient with the left cerebellum lesion, concussion is going up and down watching this, their eyes might start to skip. Their eyes might start to fail to stay on the target. Their eyes might jump around. That's a direct failure. What's the other kind of failure? Autonomic, yes, and so we might see something where the patient perfectly performs this, and an astute clinician would say, you're doing perfectly wonderful. And what else is melting down in their brain? Their autonomic system. And so what would we see? Pupils might blow out, respiration might increase, anxiety might start to rise, patient might start to tremor in a hand or the head, or they might start to see that they start to break out in a sweat, or you might start to hear their stomach start grumbling, and they might have to say, oh, I've got to go to the bathroom, and they take off. Or they start to say, I'm nauseous, or I'm dizzy. Do you have some new tools now? Very good. So there's reduced activation. This shows reduced activation in an EEG scan. This shows increased activation in an EEG scan. This shows the deeper structures we talked about, the no man's land I mentioned. That's the basal ganglia. It looks like a ram's horn. And there's the basal ganglia from the front. And here is that no man's land I talked about. The line is drawn right here. That's the tentorium. And so we have mesencephalon, pons, medulla, the brainstem, and the cerebellum. Basal ganglia is up here. And so we thank you to Sarascan for doing SPECT and PET imaging, which high-resolution technology now gives us much better care. And we can see all kinds of condition patterns like traumatic brain injury, bipolar, anxiety, stroke. We now have normative data the quality is a forensic quality. Forensic quality means you can actually testify in court on it. It's very, very scientific. We have very high-level 3D comparisons to age norms. Here's an example showing decreased activation throughout the brain. With a hot spot in the cerebellum. You can't see that on an EEG because EEG doesn't go down into no man's land, remember? I love EEG and I use it all the time, and I'm certified in it, but you don't use that all the time exclusively. You need a SPECT scan. Here's a SPECT scan including the cerebellum and those lower structures showing 3D perfusion of blood. You see some hot spots in that brain? Would those be therapeutic targets for us to do therapy on? Certainly. Here's another SPECT from above. And then this is the most important part, which I'll wrap up on, is the thalamus. You can't see the thalamus with old SPECT imaging, and you can't see it with EEG. Here's the basal ganglia, the caudate, the putamen, the globus pallidus internus and externus. These are structures that are extremely important in movement disorders. Look at the resolution. We can actually dissect them out, separate them from the rest of the brain, and see them. You can't do that with EEG. This is SPECT imaging and PET imaging. Look at how much we can dissect out overfunction versus underfunction within the thalamus and basal ganglia. Extremely important for care and recovery. So we've got a theory that talks about dysautonomia, that talks about the problems with the autonomic system, and we need to embrace that all the time as we work. There's a whole bunch of therapies that can be used for each patient. These are some of those therapies. I'm not going to go through them all. There's also some adjunctive therapies like these, EMDR and counseling and all the stuff that you guys have talked about. I won't beat them to death because you guys already had them. And there's infrared goggle exam where we have infrared goggles that can see in the dark. We put those on patients and we can see their eye movements and eye reflexes because once again, no man's land. If you don't know how to assess the lower brain, meaning the part below the tentorium, you can't treat it. You can't fix people. We can also graph this information and plot it in data and process it and show improvements. This is one of the things that we do is map and graph that information and plot it. And then I was going to do a demo, but I think this talk is more important. So I want to wrap this. Is that going back up now? Okay, then we'll forget it. Good. So thank you. Thank you. That's it. Thank you.